About the cell cycle. The mitotic phase alternates with the interphase in the cell cycle, and the eukaryotic cell cycle is regulated by different controls. So the circle represents the cell cycle, and it contains the gap 1, the S, the gap 2 phases, and the M phase. The events in the first gap is the cell grows, replicates its organelles, and the surface area over the volume gets smaller, so the cell in need to increase the surface area volume ratio for itself to obtain enough nutrients through its membrane, as well as get rid of the intense amount of waste. Before the G1 ends, there's a gap, there's a checkpoint. If the cell can not pass this checkpoint, it will go into the G0 stage. It's a non-dividing working stage or mode. Liver cells, nerve cells, and muscle cells usually here. However, the liver cells can be called back from this stage, not the neurons and the muscle cells, so far what we know. The S phase contains chromosome duplication when sister chromatids will form, and they will get attached to each other along the middle by a special protein called the cohesin. The gap 2 is what happens in here. The DNA replication gets completed, and the cell gets ready for cell division, the nucleus division, and as well as the cell division. Before the G2 ends, there's another checkpoint, checkpoint 2, G2 checkpoint. And if the cell passes that checkpoint, it will go into the M phase. If it's not, well, it has to stay in that phase. The M stands for mitosis inside of the circle, and the C stands for cytokinesis, when the cell by itself separates from just splits into different for plants and animal cells. The blue line represents the interphase that contains the first gap, the S, and the second gap. And mitosis and cytokinesis altogether will give us the mitotic phase. In the mitosis, there's also one more checkpoint to check whether the spindle fibers are okay and the sister chromatids are lined up in the metaphase plate before splitting evenly into the two newly formed cells. So these checkpoints are driven by signals. The signals can be internal signals, the amount of nutrients, amount of waste, and the size of the cell, as well as external stimuli or signals. For example, the platelet-derived growth factor. It makes the fibroblasts, a special type of cells around the injury, to proliferate, to start to reproduce more frequently. Another one is the density-dependent inhibition. When there are too many cells, well, they send out signals to each other to slow down and not to proliferate anymore. And also the anchorage dependence factor. It's kind of a touch receptor. The cell needs to have something to attach to in order to go through the cycle. Enter into S phase. So that was the G1 stuff. it goes, if the G1 is passed, it enters, encounters with the G2 phase. The G2 phase checking is the DNA synthesis completed, and is that correct? Now there is a protein, and it's a kinase, so it's doing phosphorylation, an enzyme, the cyclin dependent kinase, that it is present all around in the cell cycle in the same concentration. And when, so it is present along the cell cycle in the cell. When another protein enters into the cycle, and it's called the cyclin, its concentration in the cell varies, cycles, hence the name cyclin, going to get attached to the cyclin dependent kinase. Together they are going to form the mitosis-promoting factor. This factor gives the green light to the cell to enter into mitosis. How to turn off this signal? Well, the cyclin starts to destroy itself, or it gets disintegrated, so it's not, the mitosis promoting factor will not work anymore, because the kinase cannot work without the cyclin. Cancer cells. So the cancer cells are interesting cells that they do not obey to any of the signals. So they pass the G1, G2 and the M phases. They don't have density dependent inhibitor factor, or they do not respond to it, and they do not respond to the anchorage dependence factor either, as well as they don't do apoptosis, self-killing, because if something is wrong, cells usually do it. Also, they produce the growth factor that stimulates vessel growth into the cancer blob. And the therapy for the cancer is radiation that damages the DNA, and the cell cannot repair its DNA, so that's what the doctors count on, that they can destroy the cancer cells. Or chemotherapy like the Taxol that doesn't let the cell to go through depolymerization of the spindle microtubules, so the cell will freeze in metaphase. And that's it about the cycle.